core. These are characterized by a average diameter of 1 micrometer and with a porous with an average diameter from 5 to 25 nanometer. Then by EDX analysis, we obtained elemental mapping on this moiety of the capsule accidentally detached, and we observed a uniform distribution of silicon and oxygen, and also confirmed the presence of silver nanoparticles. After the removal of the silicon core, we obtained our core shell mesoporous structure, and we also observed a decrease of the average size. By EDX analysis, we observed the presence of silver nanoparticle inside the core and not in the shell. We also observed that the silicon core was completely removed. At this point, we want to investigate the ability of both capsules and nanoparticles as light-responsive drug delivery system. To investigate this aspect, the dialysis method was adopted. Then we dispersed the nanoparticles or the capsules in PBS solution. This solution, dialysis membrane was filled with this solution, and then it was submerged in PBS solution. Here I show you the cumulative drug release as function of time. First of all, we not observed a significant drug release for both systems without laser irradiation, the blue dots, and about the system without silver nanoparticles. Conversely, when both the nanoparticles, the black dots, and the capsules, the red dots, are irradiated by using a laser irradiation at 620 nanometer of wavelength, we can observe a release of this of our drug. For the nanoparticles, we obtain a release of 15%. For the capsule, and we obtain an amount of drug release corresponding to 22%. And in the first stage of the drug release process, we can also observe a linear correlation between the cumulative amount of drug release and the time. This demonstrates the key role of silver nanoparticles to reach a spatial tempora and the drug dosage control. At this first stage, we want also investigate the role of the physical chemical parameter. Then we want irradiate the capsule system, which showed better performance at laser wavelength closer to ones of silver nanoparticle surface plasma resonance, and we observe an increase of the drug release, and in particular a net increase was observed when we work at higher flow density. Obviously, these are only preliminary results. We are working to improve the amount of the drug release, and also we want to obtain this with this drug release under light stimulus in the near infrared region. Another important aspect about the biomedical application regards the development of multifunctional system where the diagnostic properties and the therapeutic ability are combined. In this way, great attention is focused on magnetic resonance imaging technique, which is a non-invasive technique, and this allows to detect, track, and evaluate the cell in vivo. In particular, the proton MRI allows to anatomically detected marked cells. However, the long time of acquisition, such as the possibility false positive, leads to a new strategy called a second color MRI technique, which involved the use of a second heteronuclear atoms. Among these heteronuclear atoms, more interesting is focused on fluorine 19, because the fluorine 19 is a natural abundant non-radiative isotope of fluorine. It is characterized by a geno-magnetic ratio very close to ones of proton. It is also characterized by higher sensitivity, close to ones of proton, and it's higher compared to the sensitivity of our heteronuclears. Moreover, fluorine 19 is absent in human body, is present only in teeth and in bone. Therefore, it's possible also a quantitative analysis between the correlation of the amount of fluorine 19 and intensity of the signals. Thus, to reach a good signal-noise ratio, it's necessary to increase the amount of fluorine 19. But commonly, this leads to a decrease of water solubility together with an increase of the cytotoxicity. To avoid this phenomena, the fluorinated probe must be suitable delivery in the human body by carrier system. Differently to answer to which reported in literature, we want synthesized a nanocarrier system which acts at the same time as fluorinated probe and thus drug delivery system. In this case, we selected two model drugs, dexamethasone and fluoramide. Thus, we obtained these fluorinated nanoparticles with NMR probability and with therapeutic activity. We selected the polymer, the PLGA, because the PLGA is a polymer FDA approved for biomedical application. This was functionalized with two different fluorinated amine by coupling reaction. Thus, we obtained two fluorinated polymers which contain respectively 3 and 9 fluorine equivalent atoms. This system was formulated by nanoprecipitation strategy to reach the nanoparticles, and these nanoparticles were characterized by several techniques. In particular, in the high panel, we can observe the TM image, and we evaluated the average emitter size of 80 nanometer. This data was confirmed by DLS analysis, which also provide a good PDA value. Moreover, about the NMR measurement, in both cases, we can observe the fluorine 19 signals, and this demonstrates that the self-assaying process does impair the mobility of the fluorinated chain. At the same time, we observe a net intensity of a higher intensity of the PLGA functionalized with 9 fluorine atoms compared to the which contain 3 fluoride atoms. Then we want to continue our investigation only on the PLGI functionalized with fluorine 19 atoms. Then it was loaded with the two previously selected model drugs, the dexamethasone and fluoramide, by nanoprecipitation strategy. This system was characterized by TM image. With TM analysis, we don't observe a significant increase in the average emitter size. The evaluated emitter was confirmed by the ISSN analysis, and also in this case we observe a good PDA value. Moreover, our fluorinated PLGA is able to load a higher amount of both drug compared to the not fluorinated one, and at the same time with a higher affinity towards the lefluramide. Lefluramide is characterized by higher hydrophobicity compared to the dexamethasone, and this is probably due is the process that lead to this higher drug loading effect. Moreover, about the NMR data, we can observe in both nanoparticles loaded with dexamethasone and lefluramide signals of our nanocarrier, and in the case of the lefluramide, we also observe the signals of the drug. This is an important result because in this case we could obtain a simultaneous detection of the drug and the nanocarrier. About the relaxation time, the T1 has still a good value, and conversely, we observe a decrease of the T2 value after the drug loading. This is probably due to a decrease of the mobility of the fluorinated chains, but this value is still higher compared to other value reported in literature. About the therapeutic activity, first of all, we want to demonstrate the biocompatibility of the fluorinated PLGA. These analyses were carried out on human glomerular endothelial cells and on podocytes, and no cytotoxicity was observed up to 2 mg/ml concentration. This demonstrated that the fluorination doesn't impair the biocompatibility of the polymer. Then the therapeutic activity was demonstrated on podocytes, as we can see in the image, that show a not so good morphology of cytoskeleton. After 2 days of incubation of these podocytes with our system loaded with dexamethasone, we observe a recovery of the healthy morphology, demonstrating the ability of this system like drug delivery system. In conclusion, I show you 2 light responsive nanocarrier based on silver PMA, based on the combination of silver and PMA, and we observe a better performance of the core shell structure compared to the nanoparticles. Finally, I show you 2 fluorinated PLGA nanoparticles, and we demonstrate the ability of the PLGA, which contain 19 fluorinated atoms, as an MR probe and as a drug delivery system. Currently, the investigation on its ability like MRI probe is currently in progress. Then I want to thank all people that collaborate in this research project and of the overview for your kind attention. Thank you so much.